Today's first speaker is a neuroscientist. In 2014, he won the Nobel Prize for figuring out how our brain's positioning system works. That's right, he figured out the brain's GPS. And I am excited to be able to introduce him today. So my friends, please give a warm Starmus Earth welcome to Dr. Edvard Moser.
Thanks for the introduction. Thanks to Starmus. Thanks to ESET for inviting me again. It's a pleasure to participate once again. So yesterday, we heard about challenges that are caused by human behavior. So there's nothing we would rather like than understanding human behavior. If we want to understand human behavior, we should start with the brain. And that's one of the motivations for my own work. The human brain is complex. It consists of 10 to the 11th neurons, 10 to the 15th connections, and we know that all the information lies in those connections. So where can we start? One way to start is to begin with a simple problem, fairly simple, like knowing how we find our way. How do we know where we are? How do we get from one place to the other? And that's the focus of my talk. So understanding space. Today, I will talk about space, but the space is not the space that we will hear about in many of the other talks. It's the space that is not up in the stars, but close to us, because it is a local, proximal space that we have evolved to handle. And that is what we need to deal with if we are going to survive. That includes just being able to know where you are at the moment and where other places are, how you have to move around in order to find food, how you have to move around in order to not be eating yourself, or to find a mate. All of these things are essential for any individual to survive. And you can imagine this must have evolved very early in evolution, and probably is the reason for a space system in the brain that may be common to a wide variety of species. So do we have a system in the brain for space, for knowing where we are, for knowing how to get from one place to the other? And if we have it, are we born with it? Or do we learn it? So this is not a new question. This is a question that has been around in a general form through philosophy from the earliest days. And it really came to the attention during the work of in the 1700s, where some philosophers like John Locke, George Berkeley, David Hume claimed that everything that we know is a result of sensory learning. It's a result of what comes into our brain through experience during our lifetime. So the idea is that we start out from essentially nothing. And on the other hand, at the same time, there was argued mostly by philosophers on the continent, and especially by Immanuel Kant, who argued that no, we don't start out from scratch. We actually do have ideas, thoughts, cognitions that are ingrained into our brain from the beginning. And Kant mentioned in particular our ability to perceive space, time, causality, and of course now we know there might be many others as well. It is concepts, ideas, ways of perceiving the world that come with us when we are born, we think. But is there evidence for this? Well, this is an important question of whether we actually have ideas or categories of mind that come prior to experience, can now be investigated experimentally. So this then defines one of the beginnings of psychology. So psychology in the early 1900s turned into an experimental science where you conduct experiments, you have a hypothesis, you test it, and then you revise your ideas and so on. And I wanted to emphasize the work of Edward Tolman, who was at the University of California at Berkeley, who proposed, against the common view at that time, that we actually have ways to perceive space, that we have a map in the brain, that we use both to find our way in the environment and as a framework for storing knowledge about essentially everything. He proposed there are these spatial or cognitive maps, as he called them, and he based these proposals on experiments, which he did in rats. And those rats, they are all good at navigating, so they walked around in mazes, trying to get from a starting point to an end where there used to be a treat. And he discovered, which maybe to us is obvious now, that they can make shortcuts, they can make detours, they don't always follow the same path that they once learned the first time they encountered the maze. So therefore, the suggestion from Tolman that we have maps in our brain. But if we have such maps, where are they and what are they like? So we now know that maps in the brain they cover many parts of the brain. But there are two areas of the brain that are particularly important for this, as you will understand very soon. One is called the hippocampus, which is shown in red. It's in our medial temporal lobe, and it's somewhat, actually it's not on the outside like in the figure here, but it's underneath, because that is where the cortex curves around on the middle, an old part of our cortex. And in addition, we have the entorhinal cortex, which is a part of the brain that only has been investigated the last 15, 20 years. But those two are very much connected. So what goes on inside these two brain regions? That is hard to study in the human brain, and there are many reasons. Obviously, the size, so almost 100 billion neurons, and then each neuron having approximately 10,000 connections, and those connections being so small and inaccessible that you can't access them. And of course, there are very, very few cases where you can access them in humans, essentially limited to patients who have small electrodes in their brains because for diagnostic purposes, usually in relation to epilepsy. But otherwise, then, that is not available for study in reality. So we have to go all the way to much simpler species if we want to study the brain. And nowadays, most of the work is done in mice, which are still mammals, but have very small brains, still very similar to our own brain when it comes to navigation, because navigation evolved so early. And sometimes we can go much further to the left, even to a nematode called C. elegans, with only 302 neurons, still very complex and hard to understand. But we have made some progress, and I want to highlight work in rats and mice, which started now more than 50 years ago with John O'Keefe at the University College of London, with whom we shared the prize in 2014. And what he did was that he used very, very small sensors or electrodes that can be inserted between the neurons in the brain. They are so small that they don't damage the neurons, but they are able to pick up electrical signals from the neurons. And then he asked, when are these neurons electrically active? When a rat just does whatever it wants to do, and in this case, searching around for food in a box or in a maze. So I will now start a movie. You will see a rat from the top. The rat is running around searching for chocolate crumbles, which it likes, and that's just a trick to have the rat moving around in the entire box. The box is 1 by 1 meter. And you will now listen to the electrical activity of one particular cell in the hippocampus, as was discovered by John O'Keefe. And you will hear, and you will see from dots appearing on the screen that this cell is active only when the rat is in the upper left part of the box. So each time there is a pop, 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 that means that the cell is electrically active. It fires what we call an action potential. And you already now see that this cell is active only in the upper left, so it has a certain place in the environment where it's active. And for that reason, O'Keefe called it a place cell. So this is in the hippocampus of the rat. This is shown a rat picture, rat brain picture up there, the red area. And then different cells have different activity regions. So this is shown for 10 different cells here. And this is a heat map, so red means high activity in the box, and blue means low activity. And you can see that these 10 different cells are active in different places. That means that actually the combination of neural activity at every place is unique. So that was in the 70s. Now we move more than 30 years forward in time to 2005. And this is when May-Britt Moser and I started to do work in our lab. And we recorded also from rats that were just walking in plain boxes, simple as possible, searching for chocolate crumbs around. And again, recording the activity from one particular cell. But now this cell was in the entorhinal cortex, the other area shown in blue that had almost not been explored at all at the time. And you can see that this cell is also active in certain places, but now not one place, but many places, clusters, that form a very regular pattern that covers the entire environment. And because it covers it like a grid, we call those cells grid cells. And you can see that there is a repeating unit. It's the equilateral triangles, or actually it could also be a hexagon, because the triangles can be turned two directions. And that pattern repeats all over the space. So this is like a coordinate system with 3 axes that also can be used to define locations. And if you now know that different cells have different locations, have different phases, as we say, but they also have different spacings, then you can actually imagine that anyone who reads out from that activity can actually also define locations because they are unique at every location. This is just a color code, and this is just an autocorrelogram showing that it is a repeating pattern. So now if you have populations of cells, place cells, grid cells, you can combine the activity, and you can actually read out where the rat is at any given time. So, are we born with these cells? So we started out in 2010 to record from early ages, and we found that there's some kind of grid-like pattern already then. So something is there from the very first day they explore the environment, when a rat is 15 or 16 days old. So you can see an example here. But you can also see that the pattern is really distorted and not very nice. So maybe there is a role for experience. If you then want to find out, we have to do something different. We have to record the maps not in relation to positions in the environment, but in relation to what other cells do. So you can measure maps in 2 ways. You can ask, where is a cell active relative to places in the environment, like here on the stage? Or you can ask, when is it active relative to other cells, and not care about where it is in the environment. And if you do the latter, you can do that if you have access to many, many cells at the same time. And that we now have, because there was a technological revolution a few years ago in our field, where you suddenly not only recorded activity from 5 or 6 cells at the same time, but now many thousands. And this is one example, many grid cells at the same time. In this case, there was more than 2,000 cells recorded at the same time. If you then plot the activity of each cell, here it only shows 149 of them. Each is a line of activity. When it's dark, the cell is active. When it's light, the cell is not active. Then you can ask, what is the combined activity at any given time, like in the red time slot there? And then you do that for every single time slot, and then you ask, is there any pattern to that combined activity? If those 149 cells were totally independent, you would have to plot them in a 149-dimensional coordinate system. Well, that would be really hard to see, right? You can't really see any pattern there. But since they are so highly correlated, the cells are so highly correlated, you can actually break it down to a few dimensions, for example, 3, and you can plot it again. And remember now that each dot here is one time point in the diagram at the top. And you can see then that those time points, they don't form a random structure. They actually form a very regular, they have a very regular topology, which is that of a torus, or a donut, if you want. And then, for each successive time point, they move around on that torus, as you can see on those lines. So that means that we actually have a map internally, and then we can ask, how can we quantify, how can we measure that there actually is a torus? Well, you count the holes. So if it's a torus, it's 2 holes in 1 dimension, shown there, the orthogonal, and then you have 1 hole in 2 dimensions, which is the inside of the torus. If you then count that, and just for simplicity, it's just the long lines are, with the arrows, are the very clear holes. So if in 1 dimension it's called H1, you see when a rat is moving around, when it's sleeping, 2 types of sleep, it has 2 holes in 1 dimension, and in the bottom it has 1 hole in 2 dimensions. So that means you actually have the torus there, not only when the rat is walking around, but even when it's sleeping. It is there always. So this map, this continuous map, that consists of a torus, where the activity moves around, just as when the animal walks around in the environment, activity in the brain moves around on that torus. That is our internal map. And it's very convenient that this is a continuous map, because what would happen if the map was like the Earth, like it was thought of to be like some 100 years ago? You'd come to the end of the world, and would you fall off? But with a torus, or anything continuous, that never happens. You just move around and move around and move around. So it's a very convenient way of actually generating an internal map. So then the question is this innate or is it learned? And then we come back to the very young rats. So first, this is a work of Matteo Guardamagno, who is a postdoc in our lab. So he started out first at postnatal day 15, when the grid cells are really, really distorted and messy, still not really developed the regularity. You can still see it has the 2 long lines and 1 long line as before, suggesting there's a torus. If you then go to day number 10 or 11, this is when the rats are still extremely young. They don't move at all. They're not awake. Their eyes are closed. Their ear canals are also closed. And still, you can see 2 prominent holes in 1 dimension and 1 prominent hole in 2 dimensions. And you can even go further and actually see the activity of the rat moving around on that torus when it's doing what rats do at that age, namely sleeping. So that suggests that this torus, this internal map, is there right from the beginning, as soon as you actually can record activity. But if you now go to day number 10, then there's a big change in this brain area. It changes from the activity having the pattern to the left to the activity on the right. On the left, there are, as you can see, bursts of activity. Many cells are active at the same time, and then nothing more happens in between. This is when the brain is still driven from the outside by touch or by hearing or whatever. When it goes to day 11, then the cells start to connect internally in this brain system, and then when they connect internally, you have the connections that you need for the activity to move around on a torus. And you can see it takes only hours and suddenly there. As soon as you have the internal connections, you already have the torus. So it's quite amazing how fast that is expressed. And then I'm at my final slide, and just then want to say that Immanuel Kant didn't do that bad. Actually, it was quite well in the 1700s to suggest that we actually have an internal system for handling space in our brain that we bring with us when we go to the external world the first time, and even maybe just sleep or dream about the world before we go and explore the world. It's already there, and this also means that we perceive the local space in ways that are determined by our brain, and there's no way we can escape that. The brain decides how we perceive space, and then just reminding you that Kant distinguished between das Ding für mich and das Ding an sich, and das Ding an sich, which is outside, which is independent of our brains, that we can't say anything about, at least not in terms of how we perceive things with our brain. So with that, I think I've made my point. I would just say that this system that we bring to the world is, of course, also modifiable. And one of the things the brain has to do as soon as we encounter real environments is to adapt it to the space and to use this internal template to map our locations in space as soon as we need it. But having such a template makes it go much, much faster, much simpler than you would have done without. Thank you very much.